Alright guys, I'm going to talk a little bit about a couple learning targets. We're going to go with, oops, I got through most of those. Jack drift I got through. 4 really, I'm going to go into 4. Genetic variation, maintained diploidy, disruptive stabilizing, and I'll try to get to sexual selection on natural variation. Okay, so let's take a look at first and foremost, if we could start with how genetic. Got a pen, sorry. For how genetic variations are maintained. That's slide 51. So if we look at, we talked about that one already. But if we look at ways that these things are maintained, I think slide 51 was a good one. That's adaptive. Oh, there it is. There's really 3 ways. If I can find my sickle cell picture. There's really 3 ways that things are maintained. There we go. So you got, if I go back. Nope. Let's go out. Sorry guys. I'm a mess right now. There it is. Alright. Can you write down maintaining genetic equilibrium? Or not really maintaining, but you're maintaining really genes in a population. There's 2 ways. Two ways that you can really maintain a gene in a population. And first way is what we just call diploidy. And diploidy is the idea that, if I go back to diploidy, if you just write what I underlined right here, diploidy maintains genetic variation in the form of a hidden recessive allele. So the diploidy idea is the idea that anything, this whole thing underlined right here, maintains genetic variation in the form of a hidden. So it's the idea that, like, all these genetic disorders that we keep talking about, they stay in a population because of diploidy. The idea that they can carry alleles and still not be affected. And because of that, recessive alleles will stay in the population pretty much for forever because they can't really get rid of them. Now, if you put that little example, diploidy, yeah, put the definition, but also put that, like, carriers keep genetic disorders in a population. So that helps in maintaining the genes. The next example is what we call the heterozygous advantage. So the heterozygous advantage is, there's a really great example, and it goes along with sickle cell anemia. So sickle cell, if we write this example down here, sickle cell anemia is a recessive disorder. And as a recessive disorder, the sickle cell anemia really would be lowercase s, lowercase s. If you write this down lower, this would be a person of sickle cell anemia. Okay, and for hundreds of years, if I go to our sickle cell anemia picture, for hundreds of years, people in, computer's not functioning with me here, people in these areas here, you can see the dots. The dots are malaria, and the darker circles, red, are higher percentage of the chances of sickle cell. So what had happened was these areas for hundreds and hundreds of years would get sickle cell anemia. Didn't have a great amount of treatment available. So because they, for hundreds of years, didn't have a great amount of treatment available, these people died. I'll just use blue. So these people would die. All right, but what made it different in this area, why sickle cell, the lowercase s, wouldn't go down more, is because capital S, these people did not have sickle cell. But they weren't dying, oh, I'm so sorry. They weren't dying of sickle cell anemia. These people were dying from malaria. So these people here were dying of malaria. So now you got the two outsides here are dying from two different disorders, but the evolution of this capital S, lowercase s, these people here, capital S, lowercase s, these people were actually surviving the best. And they were surviving because they were resistant to malaria. And they didn't have sickle cell. They were resistant to malaria, so they were surviving the best. So because of that, in these areas, the lowercase s got all the way up to, you see, 12.5%. It got all the way up to 12.5%, which is extremely high for a disorder that was killing people. So that's what is known as the heterozygous advantage. If you write down that the heterozygous advantage is when the heterozygous all of a sudden gains a higher fitness than both of the homozygotes. And that's what it's called. Okay, so those both help to maintain the genes in a population. They won't just allow a gene to completely be erased from a population. Good. Second part to this one, and it's talking about types of selection. Write it down. Types of population selection. And this goes along with what we talked about yesterday. What we talked about yesterday with the strawfish lab and the greens and the green. No, I'm sorry, not the greens. The blues were dying off, so the yellows were starting to. So this is type of population selection. So in this type of population selection, you got three types. One is the one that we've been talking about the most. Let's say for every one of these graphs, let's say dotted lines stand for the original population. Let's say solid lines stand for 40 generations later. Let's just use 40 generations as a generic number. So because of that, this one here is considered directional selection. And directional selection, one of the extremes is becoming more fit. So our bell-shaped curve kind of moves in one direction, okay? One of the extremes becomes more fit. Good? And that happens, I mean, frequently. You could think of a ton of different examples of where that happens. Okay, that happens with everything. It could be speed of prey, speed of predators. It happens with pretty much everything. All right. The other one, which is a little bit more rare, but sometimes you'll see disruptive selection. And in disruptive selection, if this is my original population, it could be anything. Let's say it's beak size. That's a good example. Beak size sometimes in finches works this way. If you land on one island and there's small seeds, these guys would rotate in that direction. And if they land on another area where there's a lot of large seeds, this finches would rotate in that direction. It kind of starts to make an M-like look. And in disruptive selection, that's when both extremes are successful. Oops, sorry. So that's disruptive. And eventually, what this could cause is if this one comes all the way down, that one goes all the way down, that could cause two brand new species to form. And that could cause some speciation, right? And then the last one is stabilizing. In stabilizing selection, you have the original curve, but then you also have after 40 generations, the average here starts to become more fit. So the average becomes more fit. One of the most common places you see in this one is birth mass for this one, especially if a species is kind of struggling. You'll see this one with birth mass will shrink down to a smaller window of average area because the too smalls won't survive, too small over here won't survive, and the too large over here won't be able to be born. So it starts to shrink down on that. Awesome. So that's a different learning target. The types of population selection, there's those three. And then if we could do one more, and then I'll stop talking. We could do sexual selection. So if I go to these peacocks here, we talked about this a little bit, but like, look at the difference between your peacock. You got your big tailed male peacock with all the different dances and behaviors and colorful stuff to it. And then over here, you got your drab female peacock, right? And what has caused this is sexual selection. It's the idea that females are choosing a mate based off of some characteristic. And the characteristic doesn't have to help with survival. It could help with survival, but it really doesn't have to. One of the better examples they have of this, you got, this is a good lab that a couple of environmental biologists ran, but you got the female frog, okay? The female frog usually tries to choose a male off of a longer call that it can hold for a longer amount of time. And as the female chooses the one with the longer call, the major question they want to ask is, does the longer call actually create a healthier offspring? So what they did was they took this same female, they mixed it with one with the long call, which is LC, and one with the short call, which is SC. They mixed both of her eggs with these sperms, and they wanted to test the offspring. So what they got was larval survival. The larger call was better in 95, and there's no significant difference in 96. Larval call, no significant difference in 95. A longer call was better in larval growth here, but the one that they were most impressed with was time to metamorphosis, which is really important for, especially for frogs, because if you're a tadpole, you're eaten frequently. So they were trying to show that they are better. The longer call ones are better because they had a shorter time to metamorphose to turn into a frog. So this kind of showed that, yeah, these sexual selections may seem insignificant in the survival, but maybe there is something to it where the females are choosing the healthier male to have offspring with that has the healthier children. And that's what's created so much differences. So can you write down these two? Can you write down what sexual selection is right here? Sexual selection is natural selection for mating success. And then can you also write down what sexual dimorphism is? Sexual dimorphism is marked differences between the sexes and secondary sexual selection of characteristics. Awesome. If you could hit the pause button if I went too fast and write those two down, and then we will, if you could do the Hardy-Weinberg equation, there's about 7 questions, and then that's all I got for you today. Have a good weekend. Thank you guys so much.